Okay, so I'm Tim OHara, and today I'm going to not so much talk about our voyages, although I have a few slides on that, but I want to talk about biogeography because I love biogeography. I love maps. I love, sort of imagining where all the creatures live across the planet. And that's what's really driven a lot of my science since I was a curator here in 2001. And that's a beautiful picture of a brittle star, and I'll talk lots more about brittle stars. So why did I start this? Well, when I started my job here, I had an IT background, and so I was good at databases and things like that. And I thought, how am I going to make a difference? And it struck me that at the time that really no one had any idea or hadn't really been connecting the dots in terms of large-scale marine biogeography. There was, every cruise that goes out, you can see from this little map at the top here, they sort of only cover a very limited amount of seafloor, and so you get this tiny light shone upon a small piece of the marine landscape. But really, to look at things at larger scales requires you to sort of knit all of those expeditions together to tell a bigger story about where animals live. And I saw that as a real opening and to do it through taxonomy. So by identifying animals well from all these voyages, you try and avoid the species 1, 2, and 3 thing, and actually put names on them or sort of have pseudo definitions of the species. You can sort of pull them together from various expeditions and sort of tell a big picture. And it was partly too because I was also involved in quite a few conservation initiatives, and I realised that people didn't have maps of the deep sea. So the latest UNESCO deep sea classification, which is down the bottom there, is actually based almost entirely upon biophysical surrogates, so things like organic carbon and temperature, water temperature, rather than actual biology. So people are mapping the oceans without really actually knowing what animals actually live there. And there's a reason for that. Is a lot of it, the world's a big place, and you can't, there's no way we've come up with to, I guess, remote sense the globe or the animals that live on the seafloor in a way that's sort of, we haven't come up with the technology to be able to do that remotely. And so we're still very much relying on individual samples and a lot of hard work in the laboratory. So it's really hasn't changed much probably in the last 150 years. We're still obtaining samples, putting them in museums, creating collections, creating databases, so I really wanted to put all that together and tell a big picture. In contrast, there has been maps, biogeographical maps of terrestrial and shallow water environments for a long time. Wallace, as many people would know, came up with his sort of terrestrial scheme in 1876. And amazingly, in shallow water, there was even maps as far back as 1856, based on the work of Edward Forbes, and you can see down here. And he came up with these Homozoic belts. And remarkably, I find it's remarkable how good these maps are. Like he even recognized, for example, that the Galapagos had a relatively cold water fauna compared to other places in the Eastern Pacific, and that way back in 1856, he could even see that various parts of the world had different sort of animal groups at similar latitudes. So it's extraordinary, but nothing for the deep sea. So I've kind of made it a long-term mission, and I try and get a lot of my projects, individual projects, to feed into this, is to understand, I guess, the global oceans from the point of view of one group of animals. That's the brittle stars, and I'll explain why in a minute. And it's for a lot of reasons. It's cool science, big picture, it's easy to sell. It's also has a lot of practical conservation outputs. Various management agencies, both at the national and international level, really want to know this sort of thing. So, you can get grant money, it does fund your research and that sort of stuff. So I focused on one group of animals, the brittle stars. That's a picture of another brittle star from the deep sea. And I wanted to do two things, both collect distributional data and also collect comprehensive DNA data to produce a tree of life. And so you can not only talk about patterns, but you can talk a little bit more about origins and processes. So why Ophiuroids? Well, because they're really diverse. I've toyed around with other groups as well, but I keep on coming back to Ophiuroids because they're almost present in every sample that you take. and yet they're not horrifically diverse. There's sort of right level of diversity that you don't cry on a research cruise when you just collect so many that you can't cope for the next 10 years, and nor are they so few species that you can't treat them statistically. So they're kind of in a nice sweet spot that way. And also they're becoming quite important too, because there's actually an abundant microfossil record. And previously no one really knew how to interpret a lot of those little microfossils and what they were. But now we actually know that the arm spine articulations, you can see down the end, where's my mouse? These are where the arm spines would come off the lateral arm plates. So along the animal, we've got arm segments, and on each side of the arm you get a plate and it has arm spines coming off, and the shape of these articulations are really phylogenetically informative. And so now it's opened up a whole paleo kind of sediment theme as well, that people are now looking at these and working out what past climates are and stuff based on Ophiuroid microfossils. So distributional data. I was fortunate to get a Census Marine Life grant and managed to go visit a lot of museums around the planet and combine their databases. So I've ended up with a database it's got close to 1 million specimens in it now, and it's roughly there's 2,200 described species, but I've also got about 200 well-identified undescribed species and a lot of samples as well. That doesn't mean the world is completely covered. There's, as you can see from the map there, there's a lot of the high seas in particular that are very badly covered by benthic sampling over the years. I mean, most of the research effort goes into national jurisdictions, not the high seas, which is why too that we took off very recently for the Christmas and Cocos Islands, because almost nothing is known, for example, about those two areas. And so this is on a very recent voyage. We came from Darwin and we spent 12 days researching areas around Christmas Islands until unfortunately we had to turn home, but we'll be going out again to the Cocos territories hopefully in the near future. But yes, there is definitely lots of places to still explore, particularly in the high seas. With genetic data, we started off with a transcriptome project. We did 50 transcriptomes, so that's looking at the mostly protein coding genes, and we identified about 400 that were really good, that were only single copy, that seemed to be in most species, that would look like really good candidates to build a good phylogeny. On that, we designed what's known as an exon capture process. So we actually designed these things called probes, which target individual bits of the DNA along our target exons. And so now we can regularly get about 300,000, 275 is generally our minimum of base pairs from about 425 nuclear genes and 1,500 exons. And also we also get a bit of mitochondria as well, particularly the CO1 gene. And we've done that now for 1,300 museum samples, so about 1,000 species. And so now we have a very comprehensive tree of life. It's actually produced a beautiful tree of life. Most of the branches have full support. And so now not only can we've erected a new classification for the Ophiuroids, but we can also, when we do a new survey, even if people just have CO1 or 28S data, we can actually match it to our current database and insert it in the tree of life. And this example over here is from the Clarion-Clipperton zone, which is an area in the Eastern Pacific that's going maybe mined soon for manganese nodules. And you can identify exactly where things are, how phylogenetically divergent they are. And so it opened up a lot of prospect to actually sort of characterize regional faunas as well. So let's talk about these three main axes that we'll look at here: depth, latitude, and longitude in terms of marine geography around the planet. So we'll talk about depth first, because it's the most important. As most marine biologists would know, it's actually, there's more change with depth. There's about 1,000 times greater change with depth than there is with latitude. So it's a huge amount of faunal change happens as you go from the coast all the way down to the abyss. And so this is the profile of Christmas Island where we've just been. And this is also a sort of a 3D map of the same thing. So there's Christmas Island. It looks like it's floating like some sort of thing out of Avatar here. In fact, it's the reason that's happening is because we didn't get to map the areas right close to the island because it was it wasn't safe enough for the ship to go in. So we had this sort of disconnect between the actual island itself and the bit that we mapped. Anyway. So this is roughly the profile, and I've looked at the brittle stars already and identified them, and I can see that they form bands quite clearly. So you get a set of species down to about 1100 meters, and that's largely influenced by a current that's coming through Indonesia, the Indonesian sea flow. And then you get another set of samples down to 1900, another set of species rather down to 3300, and then the abyssal fauna underneath that. And they do match quite well with water masses. So there's a lot of change happens as you go down the seamounts. And also on our abyss trip, we found very much the same thing. We didn't sample at the shallows here. We sampled from about 1,000 meters down, but you do get exactly the same. You get a depth thing. So on this ordination here, you've got all the abyssal samples and you've got all the abathal samples. This is not just Ophiuroids, by the way. This is all tectonic groups that we identified. There's a clear separation between abyssal and abathal, and then you get a regional change as well. So the samples that came from northeast Australia here, they're in the red and the orange, and then you get another set of samples down here that have a different faunal composition, and they're the blue ones there. And then actually we threw in the ones from the Great Australian Bight that CSIRO had collected as well, and they're another set of samples down here. So there's clear depth separation between bathal and abyss, and there's also regional separation as well. But we'll talk about that in a minute. In terms of DNA and the origin of this, often the distinction between shallow, as in less than 200 meters, and deep sea fauna is really old. You get whole branches on our Ophiuroid phylogeny that are really either shallow or deep. So the shallow ones are red and the deep ones are blue. So I had a student back in 2017, Lapita, who actually modelled this, and we found that there was actually quite the number of times animals actually could make the transition between the shallow and the deep were relatively few, and almost all of them had to go through an intermediate stage first. So you get shallow water species gradually expanding slightly into the deeper water before some sort of cladogenesis occurred, some sort of speciation event that separated shallow and deep species. So they go through this sort of intermediate phase first. But again, as you can see from this tree, so this is a tree of life just put into a circle here, and the center is about 270 million years. So about here is about 100 million years. And you can see some of the clades go back almost to the 200 million year mark. So it's a very ancient split between shallow and deep. And it's an old ecological pattern as well. A colleague of mine, Ben Thuy from Luxembourg, has actually looked at the fossil fauna from 140 million years ago from a rare deep sea sample, a sample that was deep sea back then. And pretty much the same families that we have nowadays were occurring there as well. So it's an old, deep, shallow difference. In terms of latitude, I did a study back in 2011, a coordinated one, that tried to actually map the distinction between the tropics and the temperate zone and the polar zones. And to do this, what we did is we took individual records and then created species habitat models by combining it with temperature and productivity and a whole lot of other things, and then model where each species occurs. We did that for 267 species, and then we could produce these sorts of maps and showing clearly that there's a tropical deep and a tropical shallow fauna, and then it goes into the temperate zone, also had a temperate deep and a temperate shallow fauna, and the temperate shallow fauna was actually divided between Australia and New Zealand. They were quite distinct. And then the polar fauna was different as well. And this is the map that I really like on the right here. It actually shows that it's not these boundaries are a bit of an artifact to the classification technique that we use to create these maps. In fact, is a very much just a gradual change. So you get a tropical fauna that gradually becomes more and more temperate as you go further south, and then you get a temperate fauna that becomes gradually more polar as you go past Macquarie Island. In terms of phylogeny, that was just based simply on actual where species occur now. So we did this big study a couple of years ago that combined both, I guess, that distributional element and a sort of a phylogenetic element as well. So this was the area we're going to be talking about for the next few slides. It's looked like a trapezium there, but it's actually shaped like that, so to preserve equal areas of the units we're looking at, like a grid, because the Earth's obviously a lot smaller near the pole, so that preserves equal areas. And then I'm going to show a lot of these sorts of charts as well. So rather than looking at latitude and longitude, it's actually more informative often to look at depth versus latitude. So 0 here is the equator, so that's up there. And this is -70 down here, which is this line down here. So that's Antarctica. So you'll always be going from the tropics down to Antarctica. And this is depth. So this is the surface, 0, and then all the way down to 4,000 meters below sea level. If you just take each species and then plot it how many species occur in each 100 meter by 1 degree cell here, you actually can see where most species are. And most species are in the tropical and temperate areas. In particular, in the shallows, there's lots of species in tropical shallows. But that sort of big pulse of biodiversity continues down below 1,000 meters. And there's even more true of the family richness. Families tend to have a bit of a wider distribution, so you get a bigger imprint here, but it's, there's more families in the tropics, both in the shallow water and in the upper Bathel. When you start adding phylogenetic data, you can sort of look at all the branch lengths and you can incorporate that into the an indice called phylogenetic diversities, and add up how much, I guess, evolutionary content you have in each cell. So a combination of all the species and how long their branches are. You can see again that's very similar. So you get the concentration of evolutionary diversity is also in the tropics. If you just look at the distribution of where all the really long stemmed animals are, so it's like this Ophiuroidea that's stuck on its own. I think that's the red one just here. It's been stuck on its own for 180 million years. So these are kind of the tuataras or the platypus of the Ophiuroid world. You can see that the major concentration of those again is in the tropics between about 200 and 1,000 meters. So that's where all the ancient things are. There's a second DNA-based analysis we did was looking at trying to actually classify all of the cells in this sort of depth by latitude plot using what's known as a phylodiversity index. I won't go into a lot of details about this. You can contact me if you're interested, but it actually incorporates again evolutionary branch length as well as distribution, species distribution in each cell, and then you can do ordinations and clusters. And then you can map those clusters, and you can see that, for example, there's a separate fauna in the tropical shallows, a separate fauna in the temperate shallows, and then you get these sort of Antarctic, temperate, and tropical zones, just like we did for the species map I showed you earlier. 9 classes turned out to, it was better in the end for us to look at 5 classes. So you can see rather than put the line through there, in the end we put a line through about here and we get 5 classes instead. And then we could build up the numbers in each class and it was easier to analyze that way. But I think really you can see our area as having 2 realms, and that's a shallow water realm and a deep sea realm. The shallow water realm only goes to the end of Tasmania, doesn't go any further than that. And there's one set of species that are quite closely related and quite diverse that live in those 2 depth by latitude plots. zones. And then there's these three deep sea ones. There's fundamentally a tropical deep sea, a temperate deep sea, and an Antarctic deep sea. And yeah, so Antarctica is very much related, even in the shallow water, it's very much related to the deep sea. In terms of, then you can take those 5 classes and you can work out which species live in each one and color code them, and you can see how they've evolved and how speciose they are. So, for example, the red is tropical shallow water species, the green is temperate shallow water species, the orange is tropical deep sea species, and blue is Antarctic species. And from these sorts of reconstructions, you can actually see how far back those lineages go. And you can plot that over time. So again, there's 270 million years in the middle of this phylogeny as you approach the end that's today, and you can take sort of concentric rings here and count the number of lineages belonging to each of those 5 groups. And what you can see here is that the tropical faunas have been around for a long time. All the really deep branches that we can confidently assign to one of those 2 groups tend to be tropical. The temperate and the Antarctic faunas are much more recent. And really that corresponds with the cooling of the Earth. So this is the Eocene, and that was the highest temperature over the last 65 million years. Back in the dinosaur era, it was also hot, and then it sort of cooled a bit in the Paleocene, it became warmer, and then gradually the Earth has cooled over time till nowadays. And so with that cooling of the Earth, it's actually, the world has become regionalized. Before, it used to be hot everywhere, and gradually it's cooled at first at the poles and then in temperate zones, and it's created new ecological niches and whole new faunas have arisen. So the Antarctic and temperate faunas are comparatively young compared to the tropical faunas. In terms of migration, that was really interesting actually, and something that we didn't expect is that in fact there's a lot of speciation occurring in the Antarctic, and it's actually been pushed into the deep sea. So rather than seeing it as a deep sea invasion of Antarctica, which might have happened originally, but certainly that's not what's happening now. There's actually a net migration from Antarctica out into the temperate, into the tropics. And there's also a lot of exchange between temperate and shallow water systems too, particularly around where those 2 systems meet. So the tropical, shallow, and the Antarctic are cradles. They're actively creating lots and lots of species at the moment. And so you can see this here. So this is, again, that's 60 million years. This is 0. So over time, the number of speciation events is still going up and up and up in Antarctica. Whereas in the tropical sea, there was a peak some time ago, back 50, 60 million years ago, and since then it's just been meandering along. So we can term the tropical deep sea a museum. There's a lot of long branches there, whereas and the Antarctic and the shallow water tropics to a lesser degree are actively creating lots of species. And the temperate deep sea tends to be a bit of a flux biome. Not so much, it's not generating many of its own species within it, but it's actually receiving species from both the tropics and the Antarctic. And also, as we'll see a bit down the track, longitudinally as well. So in terms of longitude, that's the 3rd axis. And I want to talk about a few things here. One is that we've really moved away from thinking that oceans have a continuous fauna. That's not how it happens. If you go from an Australian fauna and gradually go across the Indian Ocean to Africa, you will get a change around mid-ocean. So it gradually moves from being an Australian fauna and gradually becomes an Indian fauna on the other side. And that's true in the Atlantic as well, and also in the Indo-Pacific going towards America. Interestingly enough, that the Indo-Pacific, the division is very asymmetric. It's quite close, in fact, probably closer than this diagram shows, is quite close to the American coast. So the Indo-Pacific stretches all the way over here, but then there's actually a different fauna on the coast itself. And why has this come about? Well, there's I think, I guess, processes that are occurring over the last 65 million years. One is that there's been closures of previously open seaways, and so there's been new barriers have been created, and there's also by continents moving and pulling apart, they've also created new seaways, new dispersal zones. There are two key barriers that have occurred. Are the closing of the Tethys Sea. So this is where Africa was rammed into Arabia and effectively closed an old seaway that went from the Indo-Pacific through to the Atlantic and separating those two faunas. The more recent one, they've finally closed about 12 to 18 million years ago. The Panama Seaway finally closed about 3 million years ago, and that was a similar thing where South America and North America met, and gradually they've closed off this seaway over time and prevented animals from going through. In terms of dispersal events, the major, I guess, new dispersal route has been in the Southern Ocean. So as Africa has moved away from Antarctica and as Gondwana split up, you got currents that actually could carry animals from the Atlantic into the Pacific, and eventually too from the Pacific into the Atlantic. So as well as actually cooling the continent, these circumpolar currents, whoops, go that way, actually lead to a much cooler Antarctica as well, but they also, you can see this very much as a dispersal route. Things will from the Atlantic get caught up in these circumpolar currents and pushed into the Indian and then into the Pacific Oceans. Show you more about that. Yeah, so this is another study done by Lupita a couple of years ago, my PhD student, and she looked at shallow water barriers. So she found she got 5 different families that occurred in the shallows, and she did ancestral reconstructions about where they occurred. First, she modeled the whole shallow water system, trying to look where the major phylo clusters existed, and she identified an Indo-Pacific phylo cluster, and an Atlantic one, and then an Eastern Pacific phylo cluster as well. They were the 3 standouts. Of course, the fauna is not exactly the same in all those places. Down, for example, on the southwestern African coast, you get quite evolutionarily divergent species with longer branches, and it sort of makes it quite distinct from the other areas here. But still, these are the major clusters: the Indo-Pacific, the Atlantic, and the East Pacific. And what she was looking at mainly is when are the nodes that split, say, an Eastern Pacific from an Atlantic fauna, and where are the nodes, how far back are they that split the Atlantic fauna from the Indo-Pacific fauna when Tethys was closed. And so that's what you see down here. This is time again, this is 120 million years over here, and this is the present day. And if you look at between the east, no, I won't look at that one first, I'll look at this one first. This is Panama, so this is between the Americas and the East Pacific. So it's this one here. And the species are largely, the sister species today, and this is a fossil-based phylogeny, the calibrated, are more or less 8 million years apart. That was the average. And only a few of them actually were still splitting at 3 million years. In terms of Tethys, most of the species were splitting between 40 and 60 million years ago. So long before the actual final closure of the Tethys Seaway. And in terms of across this barrier here, it was called the East Pacific Barrier between the islands of the Central Pacific and the American coast. Only a few species have managed to actually get across and in some cases speciate, and that's happened very recently, interestingly enough. In terms of dispersal routes, the one I really want to point out is the close similarity of a lot of our baffled fauna from off Tasmania and New Zealand to Iceland. I mean, it's just extraordinary. And again, people just don't talk about this. I mean, I don't read it in the biogeographical literature, but it's really summarized by this fish, the orange ruffy, this sort of distribution where it occurs in the North Atlantic, it occurs along the East African coast, the West African coast, East Atlantic coast. And then across the Indian Ocean to Australia and New Zealand. It's more or less an L-shaped distribution, and a lot of Ophiuroids show the same thing. And I've just put a few examples here, but there's lots of others as well. Sometimes they're species pairs, so the species that occur in the Atlantic will have a sister species in Australia, but sometimes it looks like they're genetically identical. It's extraordinary. They go all the way, and they must come down through the Atlantic, and there's still gene flow then across the Indian Ocean to Australia. So there's this huge flow of biodiversity. So just putting it all together, in terms of biogeography, we've got the shallow versus deep. This is not on the chart, obviously, because it's the third dimension, but it's really important, and it's ancient. In terms of latitude, this is temperature, the different colours here, and the cooling Earth has really regionalised the fauna, creating polar, temperate, and tropical faunas. Then in terms of longitude, you've had these barriers come. We've got the two fixed barriers here, and then you've got these mid-oceanic barriers that I was showing in the chart before that have occurred as well. And they're more ephemeral things. Some things can get across, but some things are speciating across those barriers. And in terms of dispersal routes, the Southern Ocean is now really important. These Southern Ocean currents actually occur across a wide range of latitudes and have a very different biogeographical outcome. The ones that live around Antarctica are moving Antarctic species, and then the ones around where I've drawn the arrow are moving subantarctic species, but they also, some of them, because there's a Gaia here that moves around here, so its southern border also occurs from west to east, or flows from west to east, carry a temperate fauna. So you've actually got this triple transportation of species going on at different latitudes, but definitely the species are getting pushed from South Africa across Amsterdam and St. Paul Islands are there, and they tend to have a temperate fauna as well, and then they're arriving in Australia. The other one that I didn't talk about was actually during warmer times too, you can actually have gene flow through the Arctic, and there's a well-documented case in the fossil record, at least of mollusks that have been transported from the North Pacific into the Atlantic over time. So that's how I see it all fitting together. And at the end of my talk, I'd just like to thank the numerous institutions and scientists and laboratories and funding sources that I've received. Thank you.